So this is going to be the presentation for our image and EEG analysis capstone project. Our group members include Alex McConville, Danny Bowie, Kritish Bajracharya. Our affiliation is Kennesaw State University. Our professor for the course is Ying Shi, and our project sponsor is Summit Chakravarti. So our project summary, this project's purpose is to expand the scope of the existing things EEG project of analyzing the correlation of the brain's response to viewing images using EEGs. The team has researched the existing data and papers written on the Things database and Things EEG database while becoming familiar with MATLAB, EEG Lab, and Cosmo MVPA. The team's focus throughout this project will be finding the commonality between the images based on their EEG response to determine how the human brain recognizes and interprets certain images as triggers. So some of the skills that we will need for this project is the digital image processing using EEG Lab, multivariate pattern analysis, and MATLAB coding. Some of our methods for this project include the collection of EEG data, importing the raw data into EEG Lab, importing events and channel locations, EEG data set analysis, examining the raw data, filtering the raw EEG data, extracting data epochs, plotting the data, visualizing the EEG data, and selecting the useful EEG data. So some term definitions so that you can get an understanding of what features we are using in this project. The first one is going to be entropy. Entropy is a measure of the degree of randomness in an image. Also, it's a lower limit for the average coding length in bits per pixel, which can be realized by an optimum coding scheme without any loss of information. There's also going to be edge as a feature. Edges are significant local changes in an image. An edge in an image is a significant local change in the image intensity, usually associated with a discontinuity in either the image intensity or the first derivative of the image intensity. And also, as our last feature, we're going to be doing color, which is the average of the color channels, which are red, green, and blue of an image. So now Danny will go ahead and explain some of our methods and how we got to our results.
Thank you, Alex. All right, so we used cross-validation in machine learning. And cross-validation is a way to utilize machine learning models to test performance and accuracy. In the process of cross-validation, the data is split into several subsets. You'll need to train and test on separate parts of the data to create the predictive model. You must do two things with this data: train the algorithm and then actually test the model. You want to keep the two separate so you can test the model on independent data. If you train and test on the same data, you risk overfitting the model to the particular data rather than the model learning a general way to separate the two classes of interest. With cross-validation, there's a trade-off between having enough data to train on. You want as much as possible so the model can learn, and the time required to run the training multiple times. For example, you could train on the first 75% of the data set and use the remaining 25% as the test data. In our experiment, we use k-fold cross-validation, which uses the simple example. We would train on the first three subsets, test the last, record that accuracy, then rerun by training on subset 1, 2, and 4, then test subset number 3, record that accuracy. Continue to repeat this until all subsets have been tested and averaged. In this example, the data was split into 4 subsets, where in our experiment, the data was split into 12 subsets and then folded each time. This allowed us to see if the algorithm could predict the brain's ability to recognize image features based on EEG responses. As for the code, we ran image analysis using the image processing toolbox in MATLAB for the image features of edge, entropy, contrast, and color values on specific set of 200 images, which produced a CSV file. We then created a sorting program to split each feature into low, mid, and high values based on quartiles, which output into feature-specific CSV files. The cross-validation program imported the existing Cosmo MVPA data from each subject using a loop, then imported the feature-specific file. The program uses some internal Cosmo modules to function, select data for the train and test sets. We use Cosmo Match to select only the images from the feature files from the previous experiment's full list of images. This was the full list of the things database. This allowed us to slice the data and prune out any images we were not using in our test of the features. We then set the subsets to repeat 1 to 12 until all were consumed. And we can see that in this cross-validate file. Before we get into the actual cross-validation code, we must understand that the EEG records from 64 channels and electrodes across 275 time points, ranging from 100 milliseconds before the image is shown to 1 second after. Therefore, there would be a lot of noisy data in the EEG. For the classification, we use the pattern of activity across all electrodes to distinguish between the classes. The classification algorithm we used is called linear discriminant analysis, LDA. The way it works is to try and draw a boundary between the two classes based on the training data. To overcome noisy data, we use lots of trials and average across them. We then produced output into a CSV where we gathered all entropy, edge, and color to be plotted in the same graph for comparison. For the contrast was so close in values, we were not able to actually pull two different classes out of it. And for the results, let me switch back over. Go back to here. So this is a graph of the entropy, the edge, and the color. So what we know about the way the brain works is this. When we first see an image, we do not initially understand what we are seeing. It does take time for our brain to process what we are seeing, and this shows up in the EEGs as well. At 0 milliseconds, when the image first appears on the screen, the classifier performs at chance level. We start to see a response in the EEG and the accuracy around 70 milliseconds, and see the highest accuracy at first is entropy, which we can see at this blue line. This tells us that information about entropy is encoded by the brain earlier than the other features and suggests it might play an important role in object detection. Following this, information about edges and color become available. This follows what we know about the visual processing and receptive field size. The neurons which respond to fine detail and small receptive field sizes will respond first compared to neurons of large receptive field sizes. Entropy is a measure of fine detail and equates more to the small receptive field size, where edge requires larger spatial scale and will require large receptive field sizes, and therefore decoding is only possible later. Ultimately, these results are showing that information about entropy, edge, and color are present in the brain when viewing an object, and that these image features might help us in being able to recognize objects and other complex scenes very rapidly. And now I'm going to hand it over to Kritish to discuss the challenges that we faced while we were going through this project.
Thank you, Danny, for all the descriptions on the codings. Now we are talking about the challenges. Though, like we have the projects is the most important part. And this is the new technology we've been using in the MATLAB and EEG lab. We faced like frequent coding errors throughout the EEG lab on MATLAB. But Philippa, who is an PhD student from Australia, helped us working through all the errors and work together, and determining the next steps on continuing with the data. Finding out the data was also the uh, difficult part and the challenges faced by our teammates. Since we had a large amount of data, it was difficult for us to import and cluster the data, finding the commonality between the images based on their EEG responses on different images to determine how the brain recognizes and interprets certain images and responses. So this was the difficult part. And after that, clustering data on image processing, difficulty in sorting images based on the contrast, which was the most important part, because finding contrast was the main thing, and then getting the image features like color, smoothness, texture, and edge was the most challenging part, and was yeah, and finding the details on entropy and edge detection. Before it was like less to finding the less noisy, to make it less noisy, it was getting difficult. But we like working together, we faced like find the exact results. And going towards the Gantt chart. So these are the schedules we did from the beginning of the project. In the milestone one, we did images and EEG analysis, research on the data, on the EEG responses, and worked on the new coding techniques and data importing techniques. And then after we did the clustering data and create the hypothesis, testing the methods for clustering, making a group decision on the clustering data, using the methods, different methods to find the exact results. And in the final, we did final milestones and final report. We created the report and presentations. And we have here, this is the references that we get, and most of the data we have used from here. And we, like, as a whole team, we would like to give the special thank to Philippa Johnson, who is the PhD student, psychology student from Australia, was introduced by our sponsor, Mr. Sumit Chakrabarti. Philippa provided much needed information and guidance to the project. She helped us with our image processing and data processing and helped to bridge the gap between the two. She went above and beyond with providing help to us, and we cannot thank her enough. He did like two great jobs, worked together with us in each and every part, and very special thanks to her and Mr. Sumit Chakrabarti as well. Thank you.
Thank you everyone for taking the time to watch our presentation on our EEG project.